Hello friends, in this video I will explain you about the structure of MSC class 1 class 2 molecule and how they are actually process and bind with the antigen and their expression also. So let's start first with uh, the structure of MSC class 1. Here you can see it is the structure of MSC class 1 molecule. It is the general structure of MSC class 1 molecule. You can see in the left side. I will show you with the help of cursor that this is the MSC class 1 molecule, and it actually composed of a alpha chain, and this alpha chain actually composed of three immunoglobulin fold. One is here, another is here, another is here, and another chain that is called beta 2 microglobulin. And this alpha chain they are known as immunoglobulin fold because they have a structure of antiparallel beta sheet in each domain. Here you can see each domain they are composed of antiparallel beta sheet which are joined with each other by sulfide bond. And the beta 2 microglobulin actually require for the expression of class 1 MSC molecule in the cell surface, and this class 1 molecule actually present only self antigen and to the cytotoxic T cell. Okay, and here this is the typical structure of a MSC class 2 molecule. It is composed of one alpha chain. Here you can see, let's first erase this alpha chain, and this is the beta chain. The alpha chain is composed of alpha 1 domain and alpha 2 domain, and beta chain also composed of beta 1 domain and beta 2 domain. In case of MSC class 1 molecule, the antigen binding site is actually presented by the alpha 1 domain and alpha 2 domain, and they actually have a closed antigen binding site where about 8 to 10 amino acids length peptide can attach here to the peptide binding site. And in case of MSC class 2 molecule, the peptide binding site actually produced by the alpha 1 domain and the beta 1 domain, and it is actually open shape in structure, like in this way. Here the peptide actually bind, and these peptides are actually quite long, about 13 to 18 amino acids long in case of class 2 molecule. Okay, this is the biochemical structure of MSC class 1 and class 1 molecule, and this is the structure. You can see that it is here is the antigen binding site. It actually composed of anti-beta sheet, and actually the region where peptide bind is made up of alpha helix. You can see these are the alpha helix where peptide bind. This is the actually the processing pathway, how the peptide is generated within the for the MSC class 1 molecule. The here is in the middle picture you can see this one. It is the peptides protein, and the MSC class 1 molecule actually present the self antigens, self proteins, and these are the proteins come for example the cyclin which are need to degrade after the cell cycle is completed. These proteins are actually processed within a molecule known as proteasome, and within the proteasome they actually break the peptide into small fragments. Where you can see the fragments, and these fragments are now transport from the cytosol just for a second. Here I did some mistakes. I think I need to small it first. Yes, now it is good, I think. Here you can see that how the processing actually take place. Wait, sorry for the interruption. And this is the actually the endoplasmic reticulum. Here this endoplasmic reticulum. Here the MSC molecule preparation starts take place. First the alpha chain is prepared, and this alpha chain is actually attached with a chaperone protein called like colnexin, and then once the beta subunit is attached, here you can see the colnexin protein is actually released, and two other chaperone molecules known as calreticulin and tapacin is actually attached, and the peptides which are generated within the cytosol now transported into the lumen of endoplasmic reticulum via the tap molecule. This is the called tap molecule, and this is actually tap 1 and tap 2, which form heterodimer in the membrane of ER and transport peptide, and these peptides now bind with the MSC molecule, and this after binding with the MSC class 1 binding region, the tapacin and the reticulin proteins are actually released from that area. And now I will show you the general process. Here is the process mainly. This is the MSC molecule, then first chaperone bind, then it associate, then once the beta subunit comes and bind, the chaperone become release. Here you can see, then another two chaperone tapacin and tapacin and the calreticulin then binds. Then once the peptide is bind, here you can see, once the peptide is bind, these two chaperones also released, and now now once the these protein chaperones are released, now the MSC molecule actually released from the exit from the ER and exit from the ER rough endoplasmic reticulum. Here is the whole transport process of MSC class 1 molecule. Now here is the after releasing from the ER, it now enter the Golgi network actually, and then it is expressed on the membrane of the cell. But in case of class 2 molecule, it is slightly different from class 1 molecule. Here here is the class 2 molecule processing pathway. The processing actually also start in the endoplasmic reticulum, here you can see, but this peptide binding region of MSC class 2 actually need to prevent from, so that any other peptide, cell peptide, so unable to bind with that peptide binding region, and the protein chain that prevent is called invariant chain. Here you can see in yellow struck color, this invariant chain that bind, and it also help in the folding of class alpha 1 and alpha 2 and alpha 1 alpha 2 chain and alpha beta 1 beta n chain, and then the MSC class 2 molecule is transferred from the endoplasmic reticulum to Golgi network, and then in the now it is these class 2 molecules are actually engulf their antigen from outside, here you can see, then they transport that peptide via other endosomes, endomembrane system to the lysosome, where in the lysosome they beat to the low pH and other proteasomal enzyme and lysosomal enzyme, they degrade the peptide antigen into smaller fragment, and they now actually load and also the MSC molecule with cleave now interact with the lysosome. Here you can see, wait, also interact with lysosome, here you can see, and the in the lysosome the invariable chain now become also get cleaved and but a small fragment remain there and is called cleaved. And now there is another protein, actually non-classical MSC protein, that actually called HLA-DM. That HLA-DM actually replace the cleave, take the cleave and provide the antigen to the MSC molecule. And this HLA-DM also function with another molecule known as a HLA-DO. In this way, the MSC class 1 and class 2 molecules represent their antigen, and MSC class 2 molecule always represent their peptide to the class T helper cell. Thank you. If you like this video, please subscribe and put a comment. Thank you.